Hi everyone and welcome back to National 5 Biology, where we are going to be continuing on with Key Area 2 of Unit 1, and that is transport across the cell membrane. And today we are going to be looking at a different type of transport. So we've covered diffusion already. We are now moving on to osmosis. Now osmosis is similar to diffusion in that it moves from high concentration to low concentration, but osmosis is very specific. It speaks very specifically about the movement of water molecules from an area of high concentration to low concentration, but it has to be across a selectively permeable membrane. Okay, if it's not across a selectively permeable membrane, it is not osmosis. If you are asked for the definition of osmosis, it has to be that it is the movement of water molecules from an area of high concentration to low concentration, or down the concentration gradient, across a selectively permeable membrane. And that is what this looks like. So on the left hand side here, we have two solutions. Okay, so I've got solution 1 on the left and solution 2 on the right hand side. And then in the middle, we have our semi or selectively permeable membrane. They just mean the same thing. So this red dotted line in the middle is our selectively permeable membrane. And then we have a solution in here, so we've got our liquid and then we have some molecules floating about. On the left hand side, number 1, we have 1, 2, 3, 4, 5, 6, 7 molecules in here. Say this is diluting juice, okay, so we've got 7 molecules of diluting juice in a certain volume of water. On the right hand side here, number 2, we have lots more. Okay, only 7 over here, but we have way more than that over on the right hand side. If we are talking about the concentration of water, this has a very low concentration of diluting juice, which means that it is very weak juice, so it has lots of water. Okay, so this is going to be hypotonic. And hypotonic basically just means high water concentration. Over here, we have got lots of diluting juice. It's a really strong drink, which means that we don't have an awful lot of water there. Now again, osmosis is talking about the movement of water. So if we have lots of diluting juice, we have low water concentration, which we call hypertonic. So number 1 is hypotonic because it has got high water concentration. And number 2 is hypertonic because it has low water concentration. Over on the right hand side, this beaker here is what it looks like when these two solutions are isotonic. This is what would happen with osmosis. When it is osmosis, it is the water molecules that are moving. So the left hand side has high water concentration going in to low. So the water has moved from the left hand side across the membrane into the right hand side where there was low concentration. And what that has done is it has balanced out how many molecules of diluting juice we have across this. So on the right hand side, there is still more molecules of diluting juice. That hasn't changed, but the amount of water there has. So these two are now the same concentration. So they may now have ended up a different volume. The volume on the left hand side is lower than the volume on the right hand side. But because of the molecules of diluting juice dispersed amongst them, they are actually now the same concentration. And that is what osmosis is trying to achieve. It is trying to achieve both sides of that semi-permeable, selectively permeable membrane to have the exact same concentration. And when that happens, the solutions are isotonic. So isotonic just means that they are the same concentration. Now we're going to look at how that actually plays out in different cell types. So we'll start off with plant cells. So we have a cell here that is in a hypotonic solution. that means that it is in high water concentration. So say for example that this is a piece of potato. Potato is a plant, so if we've got a piece of potato, we have some plant cells there. If we were to take that potato and put it into pure water, that water on the inside of the glass, on the outside of the potato, is going to have a higher water concentration than what is inside the potato. Because potato may have water inside of it, but it's also got starch and sugar and different things in there as well. So it's not made up of pure water. So if you take something like that and put it into a pure water solution, you're setting it in a hypotonic solution. What's going to happen then is the water is going to move into that plant cell because it's going to go from high to low. So pure water is high concentration, the cell is low concentration. So placing a plant cell in a hypotonic solution means that water has moved into this and the vacuole has swollen up. In the middle here we have a normal plant cell. So the blue part is the cytoplasm, and then the white dot in the middle, that is our vacuole. So the vacuole in the hypotonic solution cell has increased in size because water has moved into it. Because water has gone from the pure water solution on the outside, which is high concentration, into the cell, which is low concentration. Because the water has moved in, the vacuole has filled up and it has swollen up and it has become turgid. Okay, that's how we would describe a plant cell placed in a hypotonic solution. It has become turgid. The cell in the isotonic solution is just a normal cell. But if we place a cell in a hypertonic solution, so say it was a concentrated salt solution, it's a solution that has got lots and lots of salt molecules inside of it. What happens then is that potato, that plant cell, actually has more water inside of it than the solution that it has been placed into. So inside of this plant cell is now high water concentration, which means water is going to leave the plant cell. Now our plant cells need water to fill up their vacuole and also for their cytoplasm. If they don't have enough water in their cytoplasm, it's actually going to start to pinch in and pull away from the cell membrane. So the cytoplasm starts to gather in the middle of the cell. And when that happens, we say that the cell is plasmolysed. To quickly recap, plant cells in a hypotonic solution are turgid. Plant cells in an isotonic solution are just normal plant cells. But then plant cells in a hypertonic solution will become plasmolysed. And now for animal cells. So the flipped a wee bit here. On the left hand side we have a hypertonic solution, which is low water concentration. So again, the water inside of this animal cell, the water concentration inside of this animal cell is higher than the solution that it has been placed in. Again, it could be a concentrated salt solution. So because that animal cell has a higher higher concentration of water, the water is going to leave the plant cell and move into the solution. That animal cell is going to shrink. An animal cell in an isotonic solution will be normal. So this is actually a red blood cell, in case we don't recognise it. And then an animal cell placed in a hypotonic solution, which is high water concentration, is going to burst. If that animal cell is taking in extra water, it is not actually capable of withstanding the water pressure. So our plant cells have a cell wall which provides extra support for them. Animal cells do not have a cell wall. And so in a hypotonic solution, they are going to burst. So to quickly summarise all of that, osmosis is the movement of water molecules from high concentration to low concentration across a selectively permeable membrane. Hypertonic means low water concentration, and the plant cells will become plasmolysed and animal cells will shrink. Hypotonic means that it is high water concentration, plant cells will become turgid, and animal cells will burst. Okay everyone, I hope that this lesson has been helpful to you whether you are revising or learning for the first time, and I will see you all in the next lesson on active transport. Take care everyone.